Assalamu alaikum. Today we want to continue talking about chapter 17, glycolysis. Now in part 1 we talked about the conversion of one molecule of glucose into two molecules of glyceraldehyde triphosphate with the net production of 2 ATP plus reducing equivalent in the form of 2 NADH. Now the final reaction of anaerobic glycolysis is the reduction of pyruvate to lactate. This is the reaction, pyruvate to lactate by lactate dehydrogenase. The reaction is an exergonic reaction. Lactate, however, is a dead end in muscle metabolism, but in the liver it can be, however, converted back to pyruvate and then to glucose via a pathway called gluconeogenesis, which we will talk about in chapter 18. Now, the enzyme that catalyzes this reaction is called lactate dehydrogenase. This is an NADH-linked dehydrogenase. It consists of 4 subunits of 2 kinds of tissue, M and H. And it has 5 possible isozymes from LDH1 to LDH5. Now, H stands for heart muscle and M stands for muscle tissue. Now, the first isozyme called LDH1 is known as H4. This one that predominates in heart muscle. It has a higher affinity for lactate as a substrate. However, the fifth isozyme called LDH5 or M4, this is found in skeletal muscle, is allosterically inhibited by pyruvate. Now, the 3 heterogeneous forms occur in blood serum. There is a very sensitive clinical test for heart disease, which depends on the existence of various forms of these isozymes in the blood. Now, the relative amounts of H4 and MH3 isozymes in blood serum increase dramatically in a heart disease known as myocardial infarction. It is heart attack compared with normal serum. So here, this enzyme can be used in the diagnosis of MI. Now, the skeletal muscle is a highly anaerobic tissue, whereas the heart is not. Of course, anaerobic metabolism does not only occur in heart and liver, but also it can occur, for example, in bacteria. The bacteria growing under the surface of the plaque, for example, must use anaerobic metabolism. Why? Because oxygen cannot diffuse readily through the waxy surface of dental plaque. Now, at this point, we might ask, what is the advantage of this anaerobic glycolysis in muscle and also the so-called alcoholic fermentation in yeast? The large advantage is that NAD plus is recycled. Now, NAD plus is the oxidizing agent which catalyzes, or which is used in the only oxidation reaction in glycolysis, must be available. If it is not, then glycolysis will stop. So in lactic acid fermentation, NAD plus, as you can see, is recycled. Now, pyruvate conversion to ethanol by alcoholic fermentation consists of 2 steps. First, decarboxylation of pyruvate to acetaldehyde. Now, the enzyme that catalyzes this step is called pyruvate decarboxylase. This enzyme requires both Mg+2 and a coenzyme called thiamine pyrophosphate. Now, if we look here, this is the enzyme and this is the reaction. Now, the vitamin B1 is the thiamine, and the active coenzyme of thiamine is called thiamine pyrophosphate. Here, notice that the thiazole ring is the active part. Now, this carbon, which lies between the nitrogen and sulfur, is a highly reactive carbon. It forms carbonyl, and this carbonyl acts as a nucleophile. It attacks the carbonyl carbon of pyruvate to form this tetrahedral intermediate, and then rearrangement of electrons, CO2 leaves, and what remains is called activated acetaldehyde. Then H plus is added here, and then a shift of electron releases acetaldehyde, regenerating the carbonyl, as you can see. The second reaction is the reduction of acetaldehyde to ethanol. Is catalyzed by an enzyme called alcohol dehydrogenase, an enzyme that catalyzes this conversion. An enzyme that catalyzes this conversion, alcohol dehydrogenase, is an NADH-linked dehydrogenase, and it is similar to lactate dehydrogenase in this property, and also both of them are tetramers of 4 subunits. And here you can see the whole reactions from pyruvate to acetaldehyde and then to ethanol. And in the muscle, we have here pyruvate to lactate. Now we want to talk about energy production of glycolysis. Now glycolysis is an exergonic process. Delta G naught prime from glucose to 2-pyruvate is equal to this amount. Now energy released in the exergonic phases of the process drives the endergonic reaction, and this is called coupling between reactions. Now some reactions are endergonic, others are exergonic, but they drive each other. Exergonic reaction drives endergonic one. Now phosphorylation is involved as 2 moles of ADP are converted to ATP. This is called substrate level phosphorylation. Now without the production of ATP to serve as a source of energy for other metabolic processes, the energy released by glycolysis would serve no purpose for any organism except to help maintain body temperature in warm-blooded animals. We saw in the previous table that the free energy changes that were listed in that, and they are under standard conditions, assuming 1 molar concentration of all solutions except hydrogen ion, which was given this value 10 to the minus 7 molar, so that the pH equals 7 and not 0. And this is in our body. Now we come to the so-called control of glycolysis. As I said before, glycolysis consists of 10 reactions. 7 of them are reversible and 3 are irreversible. Step 1 catalyzed by hexokinase, step 3 catalyzed by phosphofructokinase, and step 10 catalyzed by pyruvate kinase. These are irreversible steps, and these are the control point. These are the control points. Now, if we look at this control points in glycolysis, you see here step 1 is inhibited by glucose 6-phosphate. The enzyme hexokinase is inhibited by its own product, and this is called product inhibition. It's a control point. Step 3, the enzyme phosphofructokinase is also, as we saw, inhibited by high concentration of ATP. And the last step, pyruvate kinase, it's also inhibited by ATP, but also with other compounds, because pyruvate kinase is an allosteric enzyme. It is pyruvate kinase inhibited by ATP and alanine. Why alanine? Because alanine is the amino form of pyruvate, and they can be converted to each other by an enzyme called transaminase. So a high level of alanine indicates a high level of pyruvate, which means it would make more pyruvate can be shut down. And also it is activated by sugar phosphate called fructose 1,6-bisphosphate, so that the incoming products of the first reactions of glycolysis can be processed. Also, now pyruvate kinase itself is an allosteric enzyme, a tetramer with 3 types of subunits, M in muscle, L in liver, and A in other tissue. Is similar to lactate dehydrogenase and phosphofructokinase. Low levels of blood sugar leads to phosphorylation of pyruvate kinase, making it less reactive, and glycolysis is shut down in the liver. And we can see this here. Low blood glucose level will phosphorylate pyruvate kinase. The dephosphorylated form is more active than the phosphorylated form. So high levels of ATP inhibits pyruvate kinase, so the whole glycolytic pathway is shut down. Also, hexokinase is inhibited by high levels of glucose 6-phosphate, as I said before, and this is called product inhibition. Now in the liver, there is another enzyme called the glucokinase, has a higher Km for glucose than hexokinase, and it functions only when glucose is abundant. So if there is an excess of glucose in the liver, then glucokinase phosphorylates glucose to glucose 6-phosphate, and the purpose of this phosphorylation, of course, is the conversion to glycogen,